Assalamu alaikum, my dear students. Today we are going to talk about structure of ovule. And before talking about structure of ovule, we should be having some knowledge of the female reproductive part of flower where ovules are present. Actually, the female reproductive part of flower is known as pistil or carpel, and this pistil or carpel it is having three parts: stigma, style, and ovary. And this region represents the part where ovules or megasporangia are present. Okay, so ovules are present in the ovary. The ovules or megasporangia, they can vary in number from 1 to many depending upon the plant species. Now the details of ovule or megasporangium. Here you can see this is the detailed structure of ovule. The major portion of ovule, the bulk of ovule is composed of diploid cells, and these diploid cells are known as, they constitute nucellus. Okay, the nucellus is covered on all sides by integuments, and the number of integuments can be either one or two depending upon the plant species. The integuments cover the whole nucellus except at this region where a pore-like structure remains, and that pore-like structure is known as micropile. That structure is known as micropile. And between the cells of nucellus, in due course of time, female gametophyte formation takes place, and this female gametophyte formation or embryo sac formation, it is associated with the formation of egg cell. Okay, फीमेल गेमेटोफाइट में egg cell present होती है. Now the point, say this is the ovary wall, the point where this funiculus or stalk, it attaches the ovule to the wall of ovary. This is known as placenta. The region of ovary wall where funiculus gets attached, this is known as placenta, and the region where the funiculus gets attached to the chalaza, attached to the ovule, this is known as chalaza. Okay, so here you have funiculus. This funiculus gets attached here to placenta, ovary wall, and it also gets attached to the region of a specific region of ovule which is known as chalaza. So we have two poles in an ovule. One is known as chalazal end and another one is known as micropilar end. Okay, let me tell you this type of ovule. This is present in almost 80% of plant families. This is known as anatropous ovule. Anatropous ovule means it has turned upside down. इसका जो micropile है, micropile has come to lie next to the funiculus. Okay, this chalaza, it represents the basal region of ovule, but here you are able to see it is at the top. Reason being, this ovule has rotated, it has turned upside down, so that micropile has come to lie next to the funiculus and chalaza has assumed a top position. Okay, I will go into the details once again. We have here nucellus. This nucellus, it is composed of diploid cells. Okay, this is covered by two integuments on all sides except at this region, which forms the micropile, a small pore-like structure through which the pollen tube later on enters. Okay, and between the cells of the nucellus, female gametophyte formation takes place, and female gametophyte forms egg cell within it. Okay, the nucellus is covered by integuments. I have already talked about this. The region of the ovary wall where funiculus is attached, the region where the funiculus gets attached to the ovary wall, this is known as placenta, and the region of the funicle which gets attached to the surface of ovule, that is known as chalaza. So we have two ends, chalazal end, it is the basal end, and micropilar end, it is the top end. So this represents the detailed structure of ovule. Now we will go into the details of the chronological stages through which ovule moves. during the course of its development. First of all, it is upright, then it starts rotating, then it assumes this shape, and finally it assumes a complete anatropous shape. Anatropous shape means the micropile comes to lie next to the funicle. Okay, this type of ovule, where you can see the ovule has rotated upside down, this is present in almost 80% plant families. Okay, this represents the stages, different stages of ovule development, or you can say anatropous ovule development. Now the details of integuments. Mostly the integuments are one or two in number, and accordingly the ovules are of different types. Unitegmic ovules, they possess one integument and they are present mostly in the sympetally. Sympetally is a group of plants, okay, where you can see the petals, they are fused at the base, and this condition is present in almost 90 plant families, and the representative examples are Solanaceae and Acanthaceae. Now, bitegmic ovules, two integuments are present in these ovules. This condition is mostly present in polypetally and monocots. Polypetally represents a group of plants where the petals are free. Okay, they are free from each other, and this condition is present in almost 208 plant families. And the representative examples include Malvaceae and Brassicaceae. Then we have third condition, ategmic ovules. These ovules completely lack the integuments. This condition is of very rare occurrence, and the examples include Olex ambracata. Olex ambracata represents the condition where the ovules are ategmic. Now the origin of outer and inner integuments. The outer integument is dermal in origin, and inner integument may have either dermal or subdermal origin. Dermal origin means the outer integument arises from the epidermal cells, and subdermal means it arises from the subepidermal cells. Now there is one more important term here, which is endothecium or integumentary tapetum. Okay, in plants of sympetally, in plants belonging to sympetally group, since at the early stage of ovule development their nucellus degenerates. The nucellus of sympetally, it degenerates at an early stage of ovule development. To compensate for this early degeneration of nucellus, the innermost layer of their inner integument, it performs the nutritive function of embryo sac. It assumes a spatial function. It becomes specialized, and this specialized structure, this is known as endothecium. Okay, so endothecium is basically the innermost layer of the inner integument in case of sympetally, where the nucellus degenerates at an early stage of ovule development. This specialized structure, this performs the function of nutrition of embryo sac. It gives nutrition to the developing female gametophyte or embryo sac, and this specialized structure, this is known as endothecium. Now the functions of the integuments, it has mostly two functions. One is protection of nucellus and embryo sac, developing embryo sac, and second one is it is having nutritive function in case of sympetally. Okay, in sympetally, the innermost layer of the inner integument, it becomes it assumes the nutritive function. It provides nutrition to the developing embryo sac. Here you can see this is the outer integument. Outer integument, this is inner integument, and the innermost layer of inner integument here, it performs the function of it gives nutritive supplement to the developing embryo sac. It assumes the spatial function, and because of this spatial function, because it provides nourishment to the developing embryo sac, this is known as integumentary tapetum. Okay, integumentary tapetum means integumentary nutritive tissue. Now the details of micropile. here. Micropile is a minute opening in the ovule of a seed through which pollen tube enters. Usually the pollen tube enters through the small pore which is present in the megasporangium. In bitegmic ovules, micropiles are formed by both integuments, and that condition is known as amphistomic, or the micropile may be formed only by the inner integument, and that condition is known as endostomic, or rarely it has been found that the micropile is formed only by the outer integument, and that condition is known as exostomic. So three terms are there. One is if both the integuments are involved in the formation of the micropile, that condition is known as amphistomic. If only inner integument constitutes or forms the micropile, that condition is known as endostomic. And if only the outer integument forms the micropile, that condition is known as exostomic. Now, depending upon whether both the integuments that participate in the formation of micropile, they are straight or they are not aligned straight, the micropile can be either straight or it can assume a zigzag shape. Okay, I will repeat this. If both the integuments that participate in micropile formation, if they are straight, they will form a canal, they will form a micropile which is straight or undulating, you can say. Or if the integuments are not aligned properly, if they are not aligned straight, the micropile or the canal of the micropile, it will assume a zigzag shape. The micropile, in other words, will assume a zigzag shape, or it will form a zigzag canal. So this was all about the details of the ovule structure. In the next part, we are going to study the development of ovule, how ovule development takes place. There are different types of ovule developments. Okay. Till then, stay blessed and God bless you all.